Okay, so last time we were talking together, we were talking about different kinds of cells and the different structures they can make. And one of the things we mentioned is that some of them are able to produce capsules, right? And we said that these are like slime layers. They can be kind of like slimy and sticky. They can also be attached all over the cell wall. And when they're all over the cell wall, that's what we call an organized capsule. If they're loosely attached, just in a few points, we refer to that as a slime layer. So this is our last one that we left off with. I mentioned that these were helpful for cells to avoid dehydration. So a lot of times cells that do this are in areas that can be moist and most of the times, and then can go through a short dry period. So they use their little slime layers to protect themselves. So an example of a slime layer with an illness would be something that we would see, for example, with dental caries or with cavities. Biofilms can form on our teeth. What do we call those biofilms on our teeth? What do we go get scraped off? Starts with a T. Tartar, yeah, tartar. We go get tartar scraped off. What tartar is it's basically where we've had bacteria cells excrete these capsule-like layers and they've welded together. And so it creates like a matrix or like a mesh. If you picture like lattice work in a garden, that's kind of what they look like. And what happens when they do this is they create what biologists call a biofilm. Now this is fascinating to us because when they're in a biofilm, they actually start acting almost multicellular. They start sharing nutrients, they start protecting each other, they start communicating. So it's almost like they become like a little neighborhood. Now it's good for them because it offers more protection to them. It's bad for us because that means that now not only do we have one single capsule, now we have multiple that have welded together. And our white blood cells really don't have a chance to try to figure out how to break this down. It's like hitting a brick wall. You just don't know how to even approach it. So with this is an example, and I know it looks really bad, it looks like blood. This is actually dye. And what they did is they had a patient chew a dye capsule, and everywhere that there was tartar or a biofilm, the dye stuck to their teeth. So that gives you an example of how much tartar can slowly start to build up. This is why we have to go and get our teeth cleaned, is to remove these kinds of substances. Normal brushing alone usually isn't enough. They're very sticky and they're almost like a cement on their teeth. Okay, let's do a quick little checkpoint here. This structure protects pathogenic bacteria from phagocytosis. And I know it's been a while, huh? We have to like dust off the top ones. Okay, would that be a capsule, an endospore, a flagellum, or an axial filament?
Capsule.
Capsule, excellent, yes. So remember, phagocytosis is where a white blood cell will come up and try to eat the cell. That's what phagocytosis means. So I have a bacterium. My white blood cell comes up, grabs it, and eats it, and gets rid of it. When they have a capsule, they're so big that my white blood cell can't figure out how to bite it. It's kind of like a big hamburger. We can't figure out how to get around it. Who can give me one difference between a prokaryote and a eukaryote cell? There's a lot, so raise our hands. We'll go around the room. Yeah.
Difference is prokaryote. It doesn't have a nucleus.
Excellent. So prokaryotes do not have a nucleus. Eukaryotes do.
Does have this.
Excellent. Yes.
Enclosed organelles.
Excellent. So eukaryotes have enclosed organelles. Excellent. Now, if you're answering this on an exam, make sure you tell me which has which. Okay, like doing like a table would be good. Can we think of one other one?
Oh, you mean the same? Oh,
Just a different one. Yeah.
okay. Like prokaryotic, it's smaller.
That they have prokaryotes can grow faster. Is that what you mean?
Yeah.
Yeah. Okay. So if you're saying prokaryotes can replicate faster, they can create more cells faster. So that's true. They can replicate much faster than on eukaryotic cells. Okay. Last time we were talking, we mentioned some extracellular arrangements, and one of the ones that came up that I said we talk about later was the flagellum. And so the flagellum, and plural is flagella, the flagellum is basically an extra attachment that is going to be found outside of the cell wall of our bacteria. Now, the way that this works is some cells are going to have flagella and some aren't. If our cell is atrichous, A means without, so atrichous means without a flagellum. If our cell is able to produce a flagella, it will then be able to be motile in its environment. Now, the way the flagella works is it has three basic parts, and this flagella arrangement is different than what we see in eukaryotes. The way that they build a flagellum is very different. They have what's referred to as the filament. The filament is the whip-like tail that's going to actually be doing the swimming. So you can see the filament here is that little tail. They have the hook. The hook is what holds the tail in place so that the tail doesn't swim and then just go whoop and flip right on off. So the hook is basically holding that filament, okay? And then underneath here, we're gonna have the basal body. The basal body is like the anchor system that keeps the hook as well as the filament in place. So without those, they would fly off. So all of these are kind of built together or anchored together. Now, the way that we build a flagellum is that our cell is going to produce a special type of protein called flagellin. And what it's going to do is basically the flagellin is going to grow at the tip, and it'll keep adding to that tip so that it gets longer and longer. That way, if the flagella does get damaged or broken, it can add back on right at that tip point. So it can keep growing out. So it's not like our hair that grows from the root. Instead, it grows from the tip. So it'd be like adding hair down here. Okay, that kind of an idea. Now, this flagellum, the special protein that it produces, is very flexible. So it's able to do that whip-like motion. And it's able to be replaced if it's damaged. So if they have something happen to their little tails, they can replace them. Now, as I mentioned, the filament is that hair-like extension. That's where the flagellin is. And it's attached to that hook. And the hook is what holds the flagellin from, swimming away. The hook is made out of a different type of protein. The protein that makes up the hook is a little bit more rigid. So it can have a little flexibility, but it's not like that flagellin where it's all over the place. It's basically serving as kind of like a little core to hold everything in place. And then the basal body that the hook is attached to is an even more rigid type of protein. And that basal body is basically a protein that forms in a series of rings. So think of these as kind of like if you picture like weights on a dumbbell. That's kind of what these rings look like. So they're going to hold that hook and that flagella, the filament in place. Now, the basal body is actually going to be on the inside of that cell wall. So it's going to be on the inside of the cell wall. So it's really going to hook and hold everything in place so that we don't lose it. So if we look at this a little closer, what we're going to find is that our gram-positives and our gram-negatives have a different arrangement. And hopefully this will make sense. We'll see it up close in the next slide. The reason they have a different arrangement is because of the fact that they have different types of cell walls. And by that, I don't mean that they don't have peptidoglycan. They both have peptidoglycan. It's how they build them. Remember, one of them has a really thick cell wall. Which one has a thick cell wall? Gram-positives. Excellent. Gram-positives have a thick cell wall. Gram-negatives are very thin. So they have to anchor a little bit differently. Gram-positives, since they have that thick cell wall, they're only going to need one pair of rings. So they'll have two rings that are present. Gram-negatives, because they're so thin, they need an extra anchor. So they're going to have two pairs of rings. So we're going to see a total of 4 when we look at these. So let's see what that looks like. So here we have our gram-negative, which has that thin cell wall right here. That's that little brown layer. And here we have the gram-positive that has the thicker cell wall. Again, since the gram-positive has this thicker cell wall, it is only going to have one ring set. In a set, they come in 2. So it's going to have 2 rings total, or a set of a pair. Okay. Our gram-negatives over here, they're going to be a little different. Their cell wall is thinner. And remember, they're also going to have that outer membrane that's going to help coat the outside of the cell wall. We don't have that on gram-positives. Recall that, remember that LPS layer, that lipopolysaccharide layer that's on the outside. They're going to have one pair here and then another pair there to anchor into that LPS layer. So they have it in their cell wall as well as that external layer. So it's basically making sure that nothing falls apart from that thin cell wall. Okay, it's a little extra layer of protection. Now, when we look at our bacteria, we can see that their flagella arrangement is very different from one another. And what we find is the different types of bacteria can create different flagella. Their DNA tells them how to build it and where to put them. So, for example, I can see something that is polar, meaning the flagella is at the ends, and I can see something that's amphitrechous, which means that it's at both ends. Okay, so I have them at both ends. This is a little different than monotrichous, which means one flagella, and polar at one end. So depending on which bacterium genus I'm looking at, I will have different arrangements. So for example, this could be something like maybe we're looking at some kind of a spirochete here or a spiral bacterium. Up there, that could be something that's more along the lines of maybe like some sort of a Pseudomonas. Over here, I have Lophotrichus. That usually means I have about 8 flagella. The most common one that we see in a lot of our gram-negatives is Paratrichus, the one at the top. And that's what E. coli has. That's what a lot of our gram-negative gut bacteria have. So Proteus, things along those lines are going to have those. So remember, cells to have motility have to have flagella present or cilia present to really be able to move easily through their environment. Remember, we said some cells can look like they're moving, but that's that Brownian motion. That means that they're just bouncing around with the water molecules. So to have true movement, they need to have some sort of an external structure, either a flagella or cilia. Now, a lot of times what we see is that the bacteria will move either towards or away from a stimulus, we refer to that as a taxis, depending on what they need. So for example, some bacteria will follow or go away from light. So they will move from phototaxis meaning a light stimulant. We saw that with our pond water. When we turned our light on too bright, it's like somebody shutting a flashlight in your eyes. It's kind of like, oh, and you move away from it. They were doing the same thing. Some of them are going to go towards a food source. We would call that chemotaxis. So they're following chemicals, so they're going towards food. So depending on what kind of stimuli is in their environment, that'll affect where they go. The other thing that's interesting about our flagella is that some bacteria are going to have unique protein markers in their flagella that we can use as identification points. So basically what happens here is that our immune system builds up antibodies against proteins in the flagella themselves. And we can use this in a lab. We can do an antibody test on a bacterium with a flagella, and we can help identify what type of bacteria it is based on the proteins it has in the flagella. An example of this would be something like Salmonella. Salmonella, different strains of it produce different proteins. So I can do an antigen, we call it an antigen test on the Salmonella that I've grown, and I can tell if it's Salmonella typhi, which is going to be associated with typhoid fever, or if it's Salmonella salmonella. Well, don't you love that name? That's such an easy one, right? Salmonella salmonella, which causes salmonellosis. It's like all these salmonellas, which means that it causes tummy upset, like severe tummy upset for about 4 days. So I can tell which grouping I'm working with based on how it responds with those antigens. So another one that causes a response in our immune system is E. coli 0157. That one also has a protein that elicits a very strong immune reaction. This is why 0157 is so dangerous for us. When we get exposed to E. coli 0157, which is a special type of E. coli, what can happen is it can actually cause a toxic level to build up in our body, and it can cause organ or system shutdown. Yeah, you had a question back.
So what are the numbers 0157?
Great. So when you see something like 0157, or if you see something like flu, H1N1, those are referring to specific proteins that are present. So it's a protein marker. It's like a fingerprint. And so it's a way for us to tell where that bacterium is from and what kind of proteins it's carrying. It's kind of like if we look at people and say, okay, you're American, you're Canadian, you're Brazilian. Like we can tell kind of based on that, maybe some genes that they might have based on locality. That's what we do with the same thing here. It's just a protein marker that we use. Great question. Here is an example of how some of our Paratrichus bacteria can move. This is E. coli. If you ever watch E. coli when they're living on a slide, they do something that we call a run and tumble. They basically can fold their flagella against their body and then twirl the ends of them like a little torpedo and zoom through their environment. And then when they want to stop, what they do is they spread their flagella out. And that causes resistance, and so then they kind of roll. It's kind of like if you've ever gone snowboarding or skiing, okay, and you're going down the hill and you're doing great, and you need to stop suddenly and you sit down. And then you do this kind of roll and tumble. That's kind of what they do. It's like they put their flagella out and they just kind of roll and tumble. All right? And that allows them to stop quickly, and they can tuck their flagella and head out again. So they are very fast in their environment. Now we do have a couple of flagella out there that are very unique. And these are ones that are referred to as axial filaments or endoflagella. We see these a lot of times in spirochetes. So in certain types of these spiraled bacterium. And the way that this works is their flagella, instead of sticking out at one of the polar ends, is actually wrapped around the bacterium itself. So instead of having kind of like your ponytail hanging out, they have it like almost wrapped like a bun, like wrapped around them. If you picture Rapunzel with hair wrapped around her. Then what they do is they contract and release, and it causes them to have like a corkscrew-like motion. Now, the beauty of this is it allows them to still move very efficiently, but it also allows them to protect their flagella, because it's not just sticking out there and possibly going to get damaged. It's a little bit of an evolutionary tool for them. The problem for us is when bacteria do this, that corkscrew motion can actually be pretty damaging to our tissues. So one of the ones that does this really well is the organism that causes syphilis. We see it right here, Treponema pallidum. That's the organism that causes syphilis. And with syphilis, unfortunately, there's different stages of it. It starts as an STI, so usually in the genitalia. And for most people, that's where it'll stay, as long as they get treatment soon enough. If they don't get treatment over the next several years, the organism will start to migrate from the genitalia up through the body with this corkscrew motion, leaving damage behind as it goes. And its ultimate end result is to get into the brain tissue. That would be our third stage, syphilis. When you get to third stage syphilis, you're going to be showing some major signs. You're going to be having hallucinations. You're going to be having memory issues. If we did a brain scan, we would see basically little, bacterium in your brain cells, but we would also see scar patterns where they had spiraled through. So as you can imagine, it can lead to a lot of mental illness with the brain. There's a famous person who we suspect had syphilis who was known to not be too, stable. You guys might have heard of this person. His name was Hitler. So the medical records on Hitler indicate that when he was in his early 20s, he contracted what they referred to at that time as the gentleman's disease. That was syphilis. And as we know, as time went on, Hitler went from being a straight-A student in school, a talented artist, and somebody who wrote poetry for fun. So does that sound like the Hitler you know? You know, right? He went from being that person to being one of the most notorious, mass murderers that we've ever had on our planet. And so we highly suspect, based on what people have written in their notes, that he was hearing things, he was seeing things, he was paranoid. All of that really goes hand in hand with syphilis. So one of the things that we're trying to do is we're trying to still find parts of Hitler's remains that we can test to confirm this. They found a skull. They thought that maybe that was Hitler's. It turns out it wasn't. So it was actually a female. So based on that, then, we're still trying to find that evidence. But it's interesting because it shows us how microbes have messed with us and our history for a long time. And we're going to have a lot of those kind of incidences that we'll see in class throughout the semester. Here's another picture of that Treponema pallidum. And again, you can just see how the little axial filament, or endoflagella, the other way it's referred to, just wraps around the bacterium itself. Now, another way that our organisms can stay attached in the environment and can do a little bit of motion is through the use of what we refer to as fimbriae and pili. So fimbriae in Latin means a fringe. I always laugh, sometimes I'll get bangs and I'll call them my fimbriae because, I'll have like my forehead fringe, right? So it means a fringe. And so if you look at your bacteria, it's like they have kind of like a 70s shag going on, right? They have like these little like fringes around them. Now the difference between flagella and fimbriae is their length and the type of proteins that make them. Fimbriae do not have a basal body. They don't have flagellin. They're not super flexible. They're more rigid. Okay, so they're very different. They don't have those rings adhering them. So it's a different type of structure altogether. They start as like a little thin hair-like projection, and more and more and more can be made. And their purpose is to basically help the cell attach to another cell or the environment. They're almost like little grappling hooks. They basically like hold on when the cell's in place. So if you picture a cell, for example, in pond water, they would be able to attach to rocks so they don't get washed away by the current. If they get into our bodies, they can attach to our tissues, and it's hard to get rid of them. Our body can't just flush them out. An example would be something like Neisseria gonorrhea. So Neisseria gonorrhea is one of the organisms that we see that produces these fimbriae. And what they're able to do then is they're able to attach in the body, and we can't easily flush them or get rid of them without some sort of an antibiotic treatment. So we have to do an additional treatment. Pili are made somewhat similar to our fimbriae, but what's different is that pili will basically be built and deconstructed or broken down depending on the needs of the cell. Okay? So they're not always out. The fimbriae are always out, like a little fringe. Pili, not so much. Pili can be used to do a very limited type of motility. If we have a bacterium and it's in a situation where maybe it's getting exposed to an antibiotic or a toxin and it needs to be able to move, it will develop a pili to help it extend, grab onto something near its environment, and pull itself away. So it's not like it's swimming, it's more like a grappling hook motion. So it like shoots out this pili, grabs onto something, and then pulls itself along. As you can imagine, it's very jerky, it's not a smooth motion, and it also takes some time. So it's not something rapid like what we see with our flagella. These pili are found in pretty much every gram-negative out there. It is extremely rare to find them in gram-positives. We think it has to do with their cell walls. We think because their cell walls are thicker, it's harder for them to be able to build a pili from the cell wall. With their thinner cell walls, it's like a little projection from the cell wall. It's faster and easier for the gram-negatives. So they are something that's common in gram-negatives. I mentioned last time we were together that pili can also be used to exchange DNA. Do you remember that? Here's an example of a sex pilus, that would be singular as pilus. So a sex pilus being built from one bacterium to the next. And what's happening now is they're exchanging DNA and they're giving each other blueprints on things like how to be antibiotic resistant, how to build a capsule, survival mechanisms. We're again still not sure how the cells communicate so that one knows that the other needs this DNA. We're still not sure, we're still trying to figure that out. That's what's cool about microbiology, is it's always changing. We're always learning new things. It's like every day there's a new article out or something new to learn. Again, this would be something that we would see primarily in gram-negatives. There are a few gram-positives that can do it, but it's something that, again, we see primarily in our gram-negatives. Okay, I am a spirochete. I'm going to move by what type of method? Can I move, and you can circle all that apply. Can I move by axial filaments, endoflagella, pili, or fimbriae? We mentioned these a few slides ago. What kind of locomotion do they use? So let's look at the first one. Do you think A is correct? Do our spirochetes move by axial filaments?
Eggshell, yeah.
Yes, they move by axial filaments. Do they move by endoflagella? Yes.
So
it's A and
A and B. Axial filament and endoflagella are the same things. So they're a special type of flagellum that's wrapped around the bacterium. Okay, special type of flagellum that's wrapped around the bacterium. Pili and fimbriae, they're not really used as true locomotion. Remember, they're going to be used to help hold on to surfaces. In a pinch, We can try to use a grappling hook pili, but again, that's extremely rare. So they're not considered to be true locomotion. Pili transfer of genetic material from one cell to another would be called a what helix? What was it? It had a special name. It was on the other slide.
DNA transfer?
Let's look at our other slide here. What did I say this was called? You can see it there if you have really good vision. A sex pilus. Sex pilus. We refer to it as a sex pilus because it's actually exchanging DNA. Okay, so a special type is called a sex pilus, exchanging DNA.
I was thinking about that.
Yeah, it's a special one because a lot of times a pilus can be built as like a grappling hook, but this is a special one that's exchanging DNA. All right, let's talk a little bit about our cell wall. So our cell wall is going to be extremely important to most of our bacteria because it's going to help protect them from their environment. So first of all, the shapes that we see of our bacteria, the staph and the bacillus, that comes from the shape of their cell wall. So the shape of the bacterium tells you what their cell wall looks like. The cell wall is also going to be important to prevent osmotic pressure or lysing. So for example, if they're in an environment where there's lots of water, the cell wall is going to help make sure that those cells don't get too full of water and pop or die. Okay? It works with the plasma membrane. It's basically giving structure or rigidity to the cell. So a way to kind of resist some of that water pressure that they go through. It can also contribute to pathogenicity because we're going to see that sometimes it's harder to treat gram-negative infections than gram-positive infections because of their cell walls. Now, the structure, as we've mentioned several times up to this point, is unique to bacteria, and it's a peptidoglycan layer. So peptidoglycan means a protein and a sugar layer together. Now, the way this works is the peptidoglycan layer is basically a repeating sugar. It's a repeating disaccharide. So that means two sugar units together, repeating over and over. And then they're attached in place by a polypeptide, which is a protein, which is going to help them have some structure around them. We'll see a picture of this. Now, one thing to keep in mind, the cell wall is always going to be found outside of the plasma membrane. So the plasma membrane is what's holding the cytoplasm in place. So I go cytoplasm, plasma membrane, and then cell wall. Okay, I can't have the cell wall inside of that. Here's a picture of what our bacterial cell can look like then. So again, what we can see is that we have the cell wall in that brown layer. Around, kind of in like a purple sphere, if we picture this like 3D, we would have our plasma membrane. In white, we see that this particular bacterium has a capsule. There's ribosomes inside. There's pili inside to help them attach. A much longer flagella here that's allowing them to have true locomotion. And that would be like kind of a typical bacterial cell arrangement. Let's see if we looked at that. Now, as far as their cell wall goes, this is what their little repeating disaccharide looks like. It's a combination of two types of sugars that we refer to as NAGs and NAMs. Okay, so N-acetylglucosamine is our NAG. If I asked you about it on a test, I would put NAG. Okay, and N-acetylmuric acid is our NAM. Now, these two sugars are going to hook together, and then they're going to hook to another two sugars of NAG and NAM, and then another two sugars of NAG and NAM. So we get a layer of sugar. Okay, that's going to be building up our cell wall. We are also going to see that with our NAG and NAMs, we're also going to have some protein side chains that are going to help stabilize our sugars, and some protein or peptide bridges, remember peptide means a protein bond, that are also going to help stabilize the NAGs and NAMs as they start to stack on each other. The easiest way to think of building a cell wall is to think of building lasagna. Part of that is because I love to relate things to food whenever I can, but also because I think we've all at least seen lasagna, right? We can kind of picture that in our head. So what I want you to think of is when you see NAG and NAMs, think of the noodles of your lasagna. You know, when you're building lasagna, you add noodles, right? And then you add a meat sauce on top. Think of the meat sauce as like your proteins, and then you add another layer of noodles on top of that. Why do we have to add the meat sauce? Well, it holds our noodles in place. So that's kind of what we're gonna see when we start building our nags and nams together, is we need to have our proteins holding things in place. It'll be a nice visual to have in our head. Now, what we're gonna see is that gram-positives build their cell walls a little differently than gram-negatives. They're gonna have different components because one has a much thicker cell wall than the other. So our gram-positives, they're gonna have this nice thick layer of peptidoglycan. Yeah.
Wouldn't the gram negative has a pores that helps it to transport like pollution?
You got it. We're gonna get there. Yep, you're ahead of me. It's coming up in the next slide. Actually, 2 slides from now. But yes, you're right. So our peptidoglycans, our gram-positives, are gonna have a nice thick peptidoglycan layer here. They're also gonna have a few things to help them with stability in addition to their proteins. For example, they're going to have structures referred to as teichoic acids. These are made out of an alcohol and a phosphate. And again, their job is to help stabilize the cell wall and to regulate movement of ions in and out. Remember, ions are charged particles. So this would include things like our water molecule. A water molecule is considered to have a charge to it, right? Because of the oxygen and the hydrogen. We're also going to see that they're going to have certain types of sugars that can be present. We refer to those as surface polysaccharides. That can be unique for identification. So in other words, we can look at different sugars that can be present in some of our bacteria. Staphylococcus, for example, has a certain sugar structure. It has a mannitol that we'll talk about later. Streptococcus has a different sugar structure. So we can use these again for identification. We can do a quick test to see what kind of sugars they have. Okay, so this is in addition to the peptidoglycan. And then they also, in some species, can have another layer to this that is that waxy layer that we saw last week in lab, which is associated with our mycolic acid layer. Those were our acid-fast bacteria. Okay, and we'll see a picture of this, so don't worry. This is a unique layer that's found, for example, in the Mycobacterium tuberculosis group, the Nocardia group, which causes skin damage. We talked about those in lab, your lab book mentions those. And so those are going to be unique to pretty much those 2 genera. So let's look at what our gram-positive looks like up close. First of all, let's orient ourselves. We have NAG and NAM, so we have those present in brown and pink. We have side amino acid chains. Remember I told you those are going to help stabilize. And we have cross-bridge amino acid chains that are also going to help stabilize. So let's look at what happens. So we start to build our NAG and NAM layer. And what we can see is we have these repeating units. You can see them kind of coming out at you of NAGs and NAMs. So that's this part right here. This is the repeating units of the NAGs and NAMs. Let me see if I have a pointer in my backpack. Yeah, that or I need to start wearing like high shoes. Let's see if it'll work for me. All right. If it doesn't work right away, then I'll have to worry about it. It might be dead. I haven't charged it. Oh, okay. That's all right. I'll just try to point. Okay. So we have our NAG and NAMs here. So you can see NAGs and NAMs, pinks and browns. Okay. And they're stacked like this. Everybody with me? Like noodles and lasagna. All right. We also are going to have in the middle these protein cross bridges. So you can see the protein cross bridges that are appearing. Those are kind of like those little circles that are holding like the little dots or the marbles kind of a shape. Those are going to be the proteins in between, like our meat sauce holding everything together. Now we also have in black these other types of proteins and fats referred to as teichoic acids. Think of these as like if you stack your lasagna too high, you might put a toothpick in place to kind of like go through all the layers. Anybody guilty of that? Anybody ever made a really high lasagna, or am I the only one? I got really excited one time, made this really tall lasagna, and I had to put toothpicks in it to hold it in place. The teichoic acids, think of them as like little toothpicks. So again, they're additional stabilizers, okay? And all of these are going to work together to make the little walls of my bacterium, of my gram-positives. Now my gram-negatives are going to look very different. First of all, they're not going to have that stacking of nag and nams. So, for example, when we look here, we can see we have like 3, 4, 5, we keep stacking up. We're not going to see that with our gram-negatives. Instead, what they're going to have is they're going to have a thin peptidoglycan layer that's going to have an outer membrane around that peptidoglycan layer. That outer membrane, it can be a lipoprotein, meaning a fat in a protein. It can be a lipopolysaccharide, meaning a fat and a sugar. It just depends on the type of bacterium and what kind of cell wall they have for their DNA blueprint. Okay, so they can make different ones. We can use these lipopolysaccharide layers to help us identify some of the gram-negatives, depending on what kind of proteins they have in that layer. Again, think of it like a fingerprint. So Salmonella will have a different fingerprint than E. coli, which will have a different fingerprint than something like Proteus. Okay. Now in here, these guys are going to have a very strong negative charge, partly because of the way they're building with their polarity, but they're also going to have this barrier because of that charged layering that's going to help push away antibiotics. It's going to help push away digestive enzymes of other bacteria, and it's going to help them have an advantage when they're growing in an environment. Basically, anything that they don't like kind of gets pushed away. It's like this magnetic shield, so to speak. And so if something's polar, they can kind of control it coming close. They also have another special structure present called a porin. If you picture a golf ball, like the little dimples all over the golf ball, that's kind of what a gram-negative would look like with its porins. It has these little dimples or holes. And what these porins do is they allow the bacterium to bring in large amounts of nutrients, like larger food particles. It also allows them to bring in larger chemicals. But then it also helps them spit out things that they don't like. So when we're trying to treat a gram-negative infection, if I put an antibiotic on here, and maybe I've adjusted the polarity so I can get past their LPS layer, it'll go inside the bacterium, and they're so smart, they go, ew, I don't like this, and they spit it out through their porin. Yeah, they are so hard to treat because they are so smart. So we look at them, and look at that peptidoglycan layer. Do you see the difference already? There's only like one little line of peptidoglycan. Do you see that? So I have my nagging hams, but I only have just one line. Like I need a really skinny lasagna, one layer of noodles. Underneath again, I have the plasma membrane. I had that present in my last one. On top of my nagging hams, I now have an outer membrane. Now notice I don't have teichoic acids. The reason I don't have teichoic acids is I don't need those thicker stabilizers because my cell wall isn't so thick. So I don't need the toothpicks. Okay, I just have a single layer. I'm also going to have at the top that lipopolysaccharide layer, and then look up here in this top corner, I have a porin. So that's like a little gap, that's like a hole that can help spit things out that they don't like. Very simplistic. It is hard for us to attack these with antibiotics because they are so good at evading them. A gram-negative infection is always harder to treat than a gram-positive. So let's look at them again now side by side. So here we have our gram-positives, that thick peptidoglycan layer in purple. Here we have our gram-negatives, a thin single noodle layer. We're going to see that now we also are going to have these special proteins on the peptidoglycan layer of our gram-negative that are going to help attach to the outer membrane. On the outer membrane, we have that lipopolysaccharide, which is a fat and a sugar, which gives it a stronger negative charge, helps them control what comes in and out. They're also going to have porins present, that little X that's there, that's that little hole or that little dimple on the golf ball to spit things out. And they'll have a few proteins present to help hold things together. Over here in our gram-positives, we would have teichoic acids going through here. We have a thick peptidoglycan layer. We see a single protein, but we would have multiple protein cross bridges in here that are going to be helping hold all those layers together, like the meat sauce on the lasagna. All right? So all of these work together. This, again, is why our gram stains look so different. That thicker cell wall retains its primary dye, which makes it have that purple color. And the gram-negatives, their thin cell walls and their porins spit the dye out, which is why they don't keep that crystal violet, and we have to stain them again with safranin. Okay, so then they turn that pretty pink color. So is that making sense so far? Yeah, next.
So you would think though, because the gram positive has to be heated to allow anything to get into the cell, that it would be harder to treat than the gram negative.
Good question. Do all of them have to be treated? When we did our gram stain, do we have to heat? No. So remember, when we gram stained our staff, we have to heat fix to keep the slides and the cells together. But we didn't have to melt. And that's because most gram positives, they don't need the heat. And I'm so glad you brought that up, because that's good. Because you're putting it together in your head, and I love that. So most gram positives, they have that, the sugars, our nags and nams, but they don't have a lot of wax present or fats present. There are a few though, these guys, the atypical guys. So we're gonna have, for example, coming up in a minute, our mycobacteriums. And that's our TBs. Those are the ones we had to heat with the flame to melt open their waxy layer and then to allow it to reseal.
Okay.
So you're right. And you're absolutely right. They are hard to treat. This is why if you ever know of somebody who has tuberculosis, if they're getting treatment, does anybody know what the normal length for treatment is? I heard a whisper, yeah. 6 months to a year. And during that time, you're going to have somebody monitoring the patient because they actually switch the antibacterials every few months to make sure you don't get resistance. So they have to monitor what they're giving you because we are trying to avoid additional drug resistance. So that's a great point. You're right. Those guys evade us like crazy. So some other atypicals. Well, there's a few bacteria out there referred to as the mycoplasmas. These are different than mycobacteriums, okay, mycoplasmas. They are going to be very, very tiny. And as a matter of fact, we thought they were viruses the first time we found them. And they're so small that they've actually lost their cell wall through evolution. So mycoplasma, they only have a plasma membrane. They don't have a cell wall anymore. Now, the way that they survive is they have sterols, kind of like cholesterol around them that's protecting them. They have like a little alcohol around them. These guys, if we grow them on a petri dish, they look really odd. You know how you see bacteria on a petri dish and they kind of are raised up? I think on our slant, you can look at your slant, it's kind of like they're like a film right on the surface. When we grow mycoplasmas, they actually look like nothing's there. You don't even see them at first until you kind of tilt the plate, and then they look like dried water drops. When they grow, what they do is they burrow into the agar. They don't grow on the surface, they burrow down. They use the agar as a protective barrier. So that's one thing that they can do. Because they don't have cell walls, they can't stack on top of each other. When we want to look at them, we take the entire petri dish and put the entire petri dish, we slide it on our stage and look at it that way through the microscope. And they have kind of a fried egg appearance. Don't they look like little fried eggs here? Do you like how everything I have is food related? Do you love that? Yeah, little fried egg appearance. Okay, these guys are really unique. And like I said, they're so tiny we thought they were viruses. Where we worry about mycoplasmas usually is in the dairy industry. So mycoplasmas cause a lot of what's called mastitis, which is like an udder infection. And so it can actually cause a cow to get so swollen that they can't produce milk anymore. So this is like the bane of the dairy industry. So there's a lot of testing that's done on mycoplasmas. And they're kind of everywhere. They're so tiny, they get easily spread. They can get on your boots, they can get on your clothing. So you have to be really careful. If you ever go to a dairy or poultry farm area, you'll see that they always put covers on their boots. That's not just for the poop, it's also to make sure they're not carrying mycoplasmas in and out from one area to another. We also have the archaea. Remember I told you the archaea have a different cell wall. So the archaea, instead of having that peptidoglycan layer of nag and nams, what they have is they only have the nags in place. They don't have nams. And they only have the nag sugar, and then they attach it to a different type of protein. So we refer to their cell walls as pseudomurines. Pseudomurine just basically means that I have one type of sugar and one type of protein together. This is why the archaea are separated from the bacteria. Because the bacteria always have those two saccharides, these guys only have one. The nice thing about these walls is it gives them structure. The bad thing for us is because their walls are more porous, they're not easily gram stained. So a lot of times we can tell we're dealing with archaea if they're present because they basically turn out clear. They don't really absorb any dyes. So they don't absorb safranin, they don't absorb crystal violet, and so all you see is kind of like a grayish outline of the cell itself, kind of like how our pond water, we can see like a little gray outline of some of the cells. So how can we use this to help us? Well, the way that we can use this to help us is we can make antibiotics that target cell walls. So the beauty of this is, remember, do our cells have cell walls?
Yes. No.
No, we don't have cell walls. We have plasma membranes, but we don't have cell walls. So our first antibiotic that we ever isolated, penicillin, was able to kill Staphylococcus, because of the fact that it was a magic bullet. It would go into our bodies and not hurt our cells because it was targeting cell walls. What it actually does is when penicillin goes into our body, it attaches to the nag and NAMs as they're trying to hook together and it breaks them. So when we have a cell trying to replicate or reproduce, its offspring has cell walls that are broken that cause it to collapse. And then it weakens the nag and NAMs that are present on the mother cell and eventually cause her to collapse. Now, most antibiotics, and the reason we have to take them for several days, they're not able to kill bacteria instantly. They're stopping their offspring. That allows our immune systems to catch up. If you think about a cell that's basically creating offspring every 20 minutes, it's hard for our cells to catch up with that. I mean, they have like an army build before our cells can even get there. So if we can stop the replication, we decrease the army size, and now our white blood cells can take them out. So that's really what we're trying to do with the antibiotic, is give us an edge so that we can get ahead of them. So penicillin is going to prevent those peptide bridges. It's going to prevent nag and NAMs coming together as well. It's going to cause my lasagna to fall apart, all right? Tetracycline. Tetracycline is something that's referred to as a broad spectrum antibiotic. Have you ever heard that comment? Broad spectrum? Sometimes it's even on our pill bottles. Broad spectrum means that it's going to hurt gram negatives and gram positives equally. Tetracycline, the way it works is it targets protein formation. It targets the ribosomes. Luckily, their ribosomes are different than ours. So I can destroy their ribosomes without harming mine. What that means is my bacteria now can no longer make proteins. That's going to affect my gram negatives and my gram positives the same. So they are both going to die quickly. The problem with that, I also kill off some of my normal flora. So that's a negative. Going back to penicillin, if I'm targeting cell walls, as I mentioned when we talked about the gram stain, that means that my gram positives are much more susceptible to penicillin because they need those cell walls. If I take them away, they fall apart. Gram negatives, who we now know only have that single layer, if I take that away, they still have that LPS, that lipopolysaccharide layer, so they don't care. They keep growing. So I don't want to treat a gram negative infection with a penicillin. I would want to use a broad spectrum. Is it making sense now where we have different antibiotics for different things? Hopefully it is. Another thing that we see sometimes is we'll have what are referred to as protoplasts. Protoplasts are basically wall-less cells. So this would be something like we talked about with the mycoplasmas. These are cells that normally don't produce walls. So I wouldn't want to use penicillin to treat a protoplast because it doesn't have a wall present. Instead, I would want to use tetracycline. I want to target their proteins that they're making. Does that make sense? So I want to think about what my bacterium is as to what I'm going to prescribe. We also have some bacteria that can be referred to as L forms. An L form is basically an injured or a damaged bacterium. Think about it as a bacterium that's being treated with an antibiotic, so gram positive, that has temporarily lost its cell wall. It's on its way out. So if I was to take a patient sample and they were taking penicillin and they had a staphylococcus infection, if I took a sample a few days later, I would see staph that didn't have cell walls. Isn't that cool? That would show me that the antibiotic was working. I could tell because my staph would be weirdly shaped. Remember the cell wall gives a shape? And it would also gram stain kind of funky because it doesn't have a cell wall. It would come out more like a gram negative. Isn't that neat stuff? I mean, we can actually tell and see the antibiotic working. The difference between a protoplast and an L form then. Protoplasts never had them, never will. Like the mycoplasmas, they never had a cell wall, they don't need them, they've evolved without them. The L form, they want the cell wall, they're damaged, and if I take the antibiotic away, they will reform them. This is why we have to take antibiotics until the end of the bottle. We're all guilty of this, right? I think we're all getting better, but remember, like, you get an antibiotic prescription, you start to feel better, and what do? You stop taking the antibiotic and you squirrel it away, right? We become little squirrels. We're like putting little nuts in our cupboard. We're like, we'll use this later. The problem with that, we've created L forms, but they're not dead. So when we take the antibiotic away, they immediately come back. Now, maybe their number's low enough and we'll look out and our immune system will get rid of them. But in the microbiology world, we have a saying. You probably heard it. What doesn't kill you makes you stronger. So what happened now is we exposed bacteria to an antibiotic, and then we took it away before they died. And now what's going to happen is they're going to learn a way to get around the antibiotic if they ever see it again. They're going to develop proteins now that will neutralize that antibiotic. This is why we have so much resistance now. We've overprescribed antibiotics, we've exposed bacteria to the same antibiotic too often, and they are all able to escape it. We see this with staph. Staphylococcus aureus, I told you when penicillin was invented, it was the miracle drug, and it killed Staphylococcus aureus worldwide. We've so overused it that if I take penicillin and put it on staph now, the staph laughs at it. It gives it the middle finger and it keeps growing. It doesn't care. It grows all over the penicillin because it has adapted proteins that allow it to escape it now. What's really interesting is if you grow up a preserved specimen from the 50s, it will create this huge halo showing the penicillin still killing it. But our current staph, not at all. It'll grow over it, it won't care one bit. Okay, why are mycoplasmas resistant to antibiotics that interfere with cell wall synthesis? You can tell me.
They're protoplasts.
Yes, they're protoplasts. Excellent. They never had them and never will. So if I target a cell wall, I'm targeting something they don't have. Okay, so they never had them, never will. How do protoplasts differ from L forms? We just said what a protoplast was, right? Something that never has a cell wall. What's an L form? It produces cell walls. Excellent. It normally does. And it wants cell walls. I have temporarily removed the cell walls, but that's a temporary issue because of something I'm doing. I've created an L form. It's something that's not going to last. Excellent. Okay, let's talk about structures that are inside of our cell walls. So up to this point, we've talked about things that are outside the cell wall. We've talked about, for example, the flagella. We've talked about the pili. We have talked about the capsules, but now we're going to go inside the cell wall. So the first thing we're going to see moving inside of our cell wall is going to be the plasma membrane. This is sometimes referred to as the inner membrane. Sometimes if you read an old book, it will say the inner membrane of the cell. Now remember, I told you that we, as humans, do not have cell walls. But do we as humans have plasma membranes?
Yes.
Yes. Excellent. All cells have to have a plasma membrane. The plasma membrane is one of the most important components of a living cell. What's going to happen is this is the layer that's made of those phospholipids. Remember we talked about complex lipids? This is made of complex lipids. These are the ones that have the little polar heads and the ones that are the hydrophobic tails that are going to create a bilayer, a double layer. And the phospholipids are going to act as little bouncers of what can come in and what can go out. So basically what's going to happen is the plasma membrane is going to be the little outer membrane that holds the cell together. It's going to hold everything in place. If you think about an antibiotic capsule, how they have a coating on them to hold all the antibiotic inside, that's what the plasma membrane is doing. It's holding everything in place. It also, as I said, acts as a regulator. It controls what comes in and out of the cell. So if something harmful is trying to come in, it's going to act as an additional barrier. So this is why with antibiotics, we have to make sure that we make them in a way that gets them through the plasma membrane. So most of them are going to have a charge to them or something that you can sneak them inside. A lot of times we'll attach them to sugar molecules, so we'll pull the bacteria to think we get something good, and they'll pull it inside and then find out it's not. It's going to keep anything harmful out. It's going to have transport in two directions. So what I mean by that is I need to pull resources in, but my cell is also going to produce waste products, and I need to get rid of those. So I'm going to be able to pump things out of the cell, any kind of waste that I don't need for the cell to survive. And then the plasma membrane is also going to help with communication and identification of some of our cells, especially with our cells, because our plasma membranes rub together. We're going to find out that we have special proteins in our plasma membranes that recognize each other. It's like a self-test, which is kind of a cool analogy. So if we look at that phospholipid bilayer again, and if we recall back to what our phospholipids look like, they had that polar head and then those little fatty acid chain tails. Remember, because the head is charged or polar means with a charge, that means they like to play with other charged molecules. So remember, the heads, for example, would love to interact with water. Water is charged. The tails, which are completely filled with carbons and hydrogens, they are hydrophobic and they hate water. So they don't want to interact with water or anything that's charged. That's actually how we create kind of our bouncer layer. We have the head that goes, oh, come here, let me see you. And then the tail that goes, nope, go away. So we're able to control what comes in and out. All right. If we need to get past the tails, we're actually going to have special proteins that act as like little channels or tunnels that are going to help us get past those tails. The other thing about the phospholipid bilayer is it's really, really thin. So the phospholipids are so small. If I stacked 10,000 phospholipids on top of each other, I would have about the thickness of a piece of paper. That's 10,000 of those little guys. We're talking about two together. We're talking about a bilayer. So a very, very thin membrane, very, very tiny membrane that's present. Now, as far as the arrangement goes, again, if I put phospholipids together in any kind of medium, whether it's oil or water, they're going to naturally arrange themselves like this. They're going to form a bilayer, and their little heads are going to point out because they like to interact with things, and their little tails are going to tuck in. Think of their heads as kind of being like extroverts and their tails as being introverts. Okay, so the tails don't want to interact, they tuck in, and the heads stick out. So the heads are going to be hydrophilic, water-loving, tails are hydrophobic. Now, again, this arrangement is going to act as a barrier. If anything tries to come in that's hydrophilic, the head's going to let it pass, but it's going to hit those tails and it's going to bounce back. Non-polar or non-charged molecules will be able to move in and out much easier. So things like glucose that don't have a charge to them can be moved in and out. So other types of sugars that don't have a charge can be moved in and out. So things that are small and uncharged can be moved past. But things like water is more regulated. And if we think about this, hopefully this makes sense. We don't want to have water going willy-nilly in and out of our cell. If we did, if water just came in, whenever we had a bunch of water around, our cells would go whoop and blow up like a big balloon. And then if the water disappeared, our cells would go whoop and shrink back down, kind of like letting the air out of the balloon. That's a lot of pressure. If you picture that all day long, swelling and relaxing, swelling and relaxing. So by having this bilayer, we control the water and its flow, so we keep it as a maintained constant. Yes?
Is that for like water edema? Is that what is happening with edema?
Yep. We're having too much water retention in our cells. And so a lot of times edema also comes hand in hand with electrolyte imbalance. So with having charges being imbalanced in salts. And so we can have too high of a salt in the cell, which is pulling the water in. And so we lose our balancing technique. Excellent question. I don't know what just popped up on my screen. Hold on. It's trying to play my music for me. Isn't that nice? It's like, do you want to play a song? Yes, I do. In the middle of the lecture, I want to play a song. Okay, so this is what our cells look like in our body. We have a plasma membrane, and here's another plasma membrane. And then they're kind of interacting together. Those little plasma membranes will kind of bump into each other. But we can also have blood and nutrients and tissue layers here. But they'll be able to communicate to one another through their plasma membranes. They'll be able to kind of bond. So here's another picture of that plasma membrane. Again, just showing that phospholipid bilayer and how it's going to arrange itself. Now, when you picture the phospholipid bilayer, picture it as being viscous, kind of like olive oil. So it kind of has that fluidity to it, that motion to it, that allows the cells to kind of be flexible in their environment. And so what's going to happen then is this movement is going to give our cells kind of a little bit of fluidity. They're not going to be rigid, they're going to be flowing. Now we're also going to have other things in our plasma membrane. We're not going to have just phospholipids. We're also going to have these special proteins that are present. And these proteins are going to be present in our phospholipids as well as in bacterial phospholipids, and every cell's phospholipids. So these types of proteins are going to help our cell with survival. We're going to have peripheral proteins. These are ones that are going to be found at the outer surfaces. A lot of times these can be used to help with structural support. So they're going to make like little matrices that help our cells build a matrix or a lattice work inside. It gives them like a little framework or support. We're going to have integral proteins. These are going to be ones found inside the membrane. We'll have glycoproteins, ones that are attached to carbs, and glycolipids, which are going to be a sugar and a lipid component that allows our membranes to have some flexibility and also some oil present that allows them to slide past each other. So that way they don't get stuck. We don't want to have cells sticking together. This is what they can look like. So structural proteins, I mentioned they can hold on to like a matrix to help with the structural support. We can also have proteins that are used for communication as well as recognition. The way this works is if I have a cell pass by another cell, their proteins will gently touch each other. And if you ever see people on the campus when they see friends they haven't seen for a while, they have those complicated handshake things they do. That's basically what's happening here. Our proteins are doing a complicated handshake saying, oh yeah, you belong here. Now here's where it gets interesting. If I have a bacterial cell pass by, the proteins on its outer surface are different. So if they hit our proteins, they try to do the handshake, but it would be like me trying to do it, I'm going to mess it up. And that helps our body recognize foreign cells. Isn't that cool? It gives us a red flag. So then they can call for white blood cells to come and help them because their recognition proteins are different. This is great for things like bacteria. It's not so great for things like organ transplants. If you ever have to get a tissue transplant or an organ transplant, one of the things we worry about is rejection. Rejection occurs because those proteins in that tissue are different than yours. And so if they slide past each other, they don't know the same handshake, they're going to get targeted for destruction. This is why if you ever get an organ or tissue transplanted, you're going to have to be on immunosuppressant drugs for the rest of your life. Because we need to keep your immune system somewhat suppressed so that it doesn't attack the organ that you have. Isn't that fascinating? It'll recognize that it's not self. Yeah.
Would that same reaction be tolerant for like a breast augmentation? Or
so breast augmentation and the reactions that people have a lot of times is our bodies recognize that we have a foreign material like plastic that's inside of us. And so it causes an inflammation reaction. It's like our immune system is like, get this thing out because it's recognizing it's a foreign body. And so then you can have all sorts of side effects like swelling, headaches, general malaise. We also see that with things like shoulder implants, hip replacements, knee implants. We try to make them so it's a material that doesn't stimulate the immune system, but unfortunately some people's immune systems are more sensitive. And that's why some people will be like, oh, I didn't have a reaction at all. And then other people are like, oh, I had to get them removed. So great question. We ran into that. There's a really, if you ever want to watch like a pretty good documentary on Netflix, there's one called The Cutting Edge. And it's basically talking about how surgery has evolved over the years and how sometimes we try to use our inflammation response in ways to control things. So for example, there was a birth control device that was like a spring that they were putting in fallopian tubes. And it was basically causing inflammation or swelling and it would close the tube. The problem they ran into, and they pulled this market from the device, is it didn't just stop at the fallopian tube. It started causing big pain and inflammation in the uterus, and the stomach, and the rest of the body. It like triggered a huge immune response in some patients. So we always have to be careful with what we're putting in our bodies. So with our proteins then, those structural proteins, they're going to offer support, as I mentioned. We're going to have proteins that offer recognition. So again, they kind of slide past each other, they talk to each other. And then communication. We know that bacteria can send protein signals back and forth to each other and talk to each other, especially when they have a biofilm. Our cells do the same. For example, our T cells, which is one of our immune defense cells, one of our white blood cells that's in our bodies, it is going to circulate, and when there's signs of an infection or a bacterium present, it sends out a warning signal to all the cells in the area. It's almost like a siren call. It causes those cells to basically shut down for a little bit. And what I mean by that is usually cells are open, they're pulling nutrients in and out throughout the day. When they get a warning signal, they stop. And the idea is we don't want them pulling in bacteria by mistake. So they wait until they hear an all clear, and then they start transmitting again. Isn't that neat? Like, our bodies are fascinating with the ways that we're able to like talk to each other. And we're still trying to figure out how. Like, how are we able to communicate? They can also help with transport, because these proteins, especially the ones that are going to be integral, that cross from one side to the other, they're going to help us with moving materials across those tails that don't like anybody, the little introverts, right? So for example, if I have a charged molecule like water, I still need to be able to move it in and out of the cell. So I can move it through these special tunnels, there are special protein channels that are open and close as I need to move the water, and allow me to have a way to get past those tails that would otherwise reject it. So anything that's polar can move across that way. So these protein molecules, again, they can be arranged in different patterns. No two proteins are alike. Our cells are all a little different from each other. They have the same general format and structure and function, but it's just like our DNA. We all have a little variety to them. Those peripheral proteins, as I mentioned, they help acting like a support. And those integral proteins are the ones I just mentioned that are basically going to act like a little channel to move nutrients in and out. And so these would be things like our polar or our charged molecules that otherwise would hit those tails and would bounce back. Now, the beauty of this then is these proteins in the plasma membrane, they help it stay strong and they help it stay regulated. So all of these work together to make our plasma membrane as a whole. So with this then, the plasma membrane and its proteins are able to protect the cell. They're able to have a little lubrication present so cells can pass by each other without damage. And they're able to communicate with one another if something's wrong. Now, we have some bacteria out there that they'll produce waste products, we refer to these as toxins, that can target our protein surfaces on our plasma membrane. So for example, things like cholera or botulism, these are going to be associated with diseases that will cause intestinal upset. And the reason that they're causing an intestinal upset is the toxin from the bacteria are attaching to our intestinal cell lining. It's causing an inflammation response. And our bodies, when something's irritating them in the intestines, they hit the easy button, the eject button, and so then you end up having massive diarrhea. If they're really upset, then they'll send a signal to the brain and you'll start having nausea as well and start vomiting, because it's like they're trying to clear your body out. It turns out that a lot of times with these organisms, our immune reactions are actually the disease. They're actually worse than any kind of illness the bacteria itself can cause. It's like we overreact. That's especially true with cholera, which we'll talk about in a few chapters. So when we put the plasma membrane together with its proteins and its phospholipid bilayer, we refer to it as a fluid mosaic model. Isn't that lovely? Remember, a mosaic are kind of like those paintings you sometimes see in the mall, where from a distance you'll see the face of a famous person, and then when you get close, it's a whole bunch of little pictures. Have you ever seen that? That's what a mosaic means. It means little pieces making a whole. So we have little pieces, we have little proteins, we have little phospholipids that together make a whole. And they are going to be able to control what comes in and out, so they can control things like water and oxygen, sugars, any of those things can be regulated through that plasma membrane. Now, the plasma membrane can be damaged. We have to be careful. For example, certain types of cleaners out there will use alcohols or quaternary ammoniums to damage the plasma membrane of the cell. If that's the case, it can cause damage in the cell that can become unstable. We have to be careful with these types of cleaners. There are certain cleaners out there that will tell you to wear gloves when using them. The reason for that is because if they're impacting the bacteria's plasma membranes, they're going to impact ours as well. Okay, so that's why certain cleaners, especially like certain hospital cleaners that are very strong, the quaternary ammonia ones, they're going to tell you to wear gloves when you're using them, or to wear a mask and gloves, so that you prevent yourself from inhaling them. Antibiotics that target the cell wall are safe for us though, because we don't have cell walls. So as long as we are targeting a cell wall, we're good. If we're targeting a plasma membrane, we got to be careful, because that means that can hurt us. So again, we always just have to think about what we're using. Now, the plasma membrane can also control the materials that flow in and out. So materials move across the PM, that PM stands for plasma membrane. And materials can flow either passively, they can flow through facilitated diffusion, or they can flow through active diffusion. Diffusion is where I'm looking at the flow of a molecule or a concentrate. Okay? So the way that this works is the different types of diffusion will help show me what kind of movement it's going to have in the cell. I can have passive diffusion. Passive diffusion is where I'm moving from a high to a low concentration, like a balanced concentration. An example of this would be like if I have a beaker of water and I put a drop of red dye in there. The red dye, when it first goes in, is super concentrated. It's high in that area. And what's going to happen is it will diffuse out. It will try to balance by moving out. And so it moves from high, concentrated, to low, to be in an area that's less concentrated. That's passive. It's basically moving down a gradient flow. Think of like sliding down a slide. We're moving from the top to the bottom. Okay, it's a nice flow. So there's a picture of my dye molecules where I first add them, and then they flow out. Now, other types of diffusion that can occur. A lot of my substances can do this passive diffusion, moving from high to low. That could be things that are going to be like my simple sugars, moving from high to low. I run into a problem though when I try to move my polar molecules, ones with a charge. They try to cross the plasma membrane, they hit those hydrophobic tails, and they can't get past. So now I can't just move high to low. I have to do something else. So for a lot of those ions, I have what's called facilitated passive diffusion. Facilitated passive diffusion means I'm still moving high to low, but I have to use a protein helper or a channel to help me move. Okay? So if I try to move from high to low, from outside the cell into the cell, I hit that barrier and I can't do it. So now if I have a special protein present, I can cross through the protein and do it. That's facilitated. I need a helper. I'm still moving high to low though. Okay, so the flow is the same. This is different than active diffusion. Active diffusion means instead of going down my slide from high to low, I'm pushing back up the slide and going from low to high. Active diffusion means I'm going the wrong way. So I'm going to have to pay a price to do that. That means I'm going to have to use an ATP molecule. Just like if we're going to climb up a slide, it's going to take more energy than sliding down, right? We can sit on a slide and just let go and we're going to be at the bottom. But if I want to turn around and climb up it, I use energy. Same idea with the active diffusion. So, for example, facilitated diffusion has a protein channel moving high to low. Active diffusion is where I'm pumping against my gradient. I'm going low to high, which means I have to use energy. Now, the reason I go low to high is a lot of times it's like a prep step for something that might occur later. If you've taken physiology, you might have heard of like sodium potassium pumps. and how our cells will pump sodium and potassium against their gradients to allow a quick switch so that it can allow a quick muscle movement. So sometimes I have to have things pumped against their gradient. So that's going to be an active diffusion. We have enough time to get into osmosis, and then we'll stop with osmosis. Osmosis is going to be a similar process to diffusion, but the difference with osmosis, in diffusion we were looking at the solutes, the sugars, the glucoses, the oxygens. With osmosis, we're looking at the flow of water. So we're looking at only the water across the plasma membrane. And so the osmotic flow or osmotic pressure is basically going to be balanced by that plasma membrane. And we're going to have different situations that our cell is going to be exposed to. So what's going to happen is if I have a cell, for example, that's in a process or in a solution of water that has more solutes out here, maybe more salts, my water is going to flow differently. So if I have something where I have more salts inside, the water is going to flow in to try to dilute out or balance those salts. If I run into a situation where I have more salt outside, my water might flow out. You guys have probably heard of this before through the terms hypertonic, hypotonic, and isotonic. The difference is it's all about the flow of water. So let's look at a better picture. Here I have a cell, and it has a set number of solutes inside and a set number of solutes outside. The water in the cell is perfectly balanced with the solutes inside and out. And so what that means is my water is not going to move. So isotonic means that I'm in balance. I'm stabilized. That's what my iso means. Hypotonic. Hypotonic means that I'm going to have a situation where I have more salts present in my cells than outside. Maybe this is something like I drank DI water. Are we supposed to drink super distilled water? No. What that means is we've removed charged particles from it. So when we drink it, it's ultra pure. That means there's no solutes. So if I drink that water, I'm drinking in ultra pure water, which means I have more salt in the cell, which means the water is going to flow into the cell to try to balance out the salts that are inside. Make sense? That's going to cause my cell to balloon up. Okay? Hypertonic is the exact opposite. This would be like if I drank salt water. When I was learning how to surf, I drank a lot of salt water. I thought I was going to die. And it was like awful. Felt bad. But what happened is when I drank that salt water, not on purpose, just getting tumbled, that salt water would go in my body and there's more salt around my cells than inside. And the water in my cells would flow out, trying to balance it. An easy way to remember these is to keep track. When you see hypertonic, think of E for exit. What that means is the water is exiting, okay? When you see hypotonic, take this first part, this hypo, and turn it into hippos. Do hippos like water? Are hippos big? Yeah, so your cell is getting big, it's swelling up like a hippo. Okay, that's just an easy, simple way to remember it. So what this will do to our cells, and this is the last slide, I promise. What this will do to our cells then is if I'm in a situation that is going to be hypertonic, meaning that water is leaving, it can cause my cell to shrivel up, and it can, for example, cause my plants to wilt. It can cause my body to feel dehydrated because I've lost water there. If I'm in a balanced situation, nothing changes. If I'm in a situation where I drink ultra pure water, I'm in trouble because now I'm blowing up like a hippo, and it's causing pressure on my cells. It also is causing my plants. If you ever water them with super DI water, they go and they like blow up. It's like really sad. And then eventually it kills them because of the cell stress. We had a lab tech who loved to use our ultra pure water to water her plants because she said they looked happier. They weren't happier. they eventually died, within the next time. Okay, we'll stop here. So what that means is our exam will cover through osmosis. The rest of the stuff will be on the next exam. All right, so you do not have to study anything that we haven't covered. All right? All right, so we'll stop here. I went a few minutes over, so if you guys want, we can start lab at, do you guys want to start at 10:35?